Our third speaker is Jiongming Wang, graduate student in biostatistics, who will be speaking on investigating causal relationship between blood-based biomarkers and the risk of psychiatric disorders.
Okay, so now for today, I'm going to talk about our current work on investigating the causal relationship between blood-based biomarkers and the risk of psychiatric disorders. To start with, in 2020, about 50 million adults in the US were estimated to live with mental disorders, which accounts for about 1/5 of the US adult populations. And so far, the mental disorder has been one of the leading causes of the disorders in the US or worldwide over the past decades. And it has been posing great challenges to both people's well-being and public health systems. So the investigation of the relationship between the psychiatric disorders, together with some possible informative biomarkers, can inform us about the disease etiologies and further to improve our drug research and development in broadly mental disorders. Actually, over the past decade, there are many studies have been identified several the biomarkers have the significant association with the psychiatric disorder. So here I illustrate one example for the major depressive disorders. However, whether this kind of association is causal relationship still remains unknown. So in our current study, we wanted to investigate the causal relationship between the blood-based biomarkers and the risk of mental disorders. To be more specific, in this project, we investigate the blood-based traits of interest, including the basophil, eosinophil, lymphocyte, monocyte, neutrophil, and platelet count. And for the mental disorders, we investigate the major 4 kinds of disorders here. They are major depressive disorder, bipolar disorder, anxiety disorder, and PTSD. So, in order to infer the causality, we apply the Mendelian randomization as our primary method on the Michigan Medicine Electronic Health Record linked with the MGI dataset. Here is our analysis protocol. So we started from the MGI phase 4 cohort, and I think the MGI team has done very comprehensive quality control for the genetic data, so it facilitates us to focus on the unrelated European samples in the phase 4 MGI samples. And then for the phenotyping, regarding the electronic health data phenotyping, we use the ICD-10 code to phenotype the psychiatric disorders, and we use the low-in code to phenotype the lab test traits here. And then we link this genetic data and the EHA data together, and then we do the GWAS analysis and the downstream one-sample Mendelian randomization together with the following sensitivity check and the sanity check to assess their pairwise causal relationship between these 2 kits. Okay, so here comes the result. This is our univariable Mendelian randomization result, and here we found 5 types of significant relationships from the blood-based traits to the psychiatric disorders. After controlling some known pleiotropy variants, there were two causal relationships were replicated in our multivariable Mendelian randomizations. They are the increase in the platelet is causally associated with the increasing risk of major depressive disorders and anxiety disorders. And in addition, we also decreased 2 additional causal relationships in this multivariable MR analysis, that the decrease in the basophil and the decrease in the monocyte will increase the risk of major depressive disorder and bipolar disorder, respectively. So to sum up, we have identified several causal relationships between blood cells and the mental disorders, and some blood cells, like we have shown before, like the platelet, have the causal effects on the multiple mental disorders, while the others, like the monocyte or the basophil, are causally associated with one of the specific disorders. And this kind of differentiated effect may be a signal of the similarity and differences with respect to the etiologies for the different types of psychiatric disorders and the metabolism pathways. And we are now assessing this reproducibility through some external cohorts and summary studies as our future works. Thank you.
Questions?
A couple of questions.
Yes.
On what basis are you able to say that there's a causal relationship between your observation and major depressive disorder? And then two, are there any differences in the observation between sexes, say male, females? And the reason why I ask that is because the association identified with platelets. Platelets, there are some studies that have revealed that platelets can actually secrete testosterone, for example. So are there any differences in sex? And also if you have any observation on testosterone in some of the factors that you looked at?
I think that's really like good comments here. So we adjust for the sex and like age as normal, like in the GWAS analysis. But yes, we are like for some traits we can. I did not show here. Like especially for the red blood cells traits, we may have some differences. So yes, I'll take it into account in the future. Thank you.
Time for one more short question.
Does the MGI data have contain 2 time points so you could verify your first finding?
Yes, absolutely. In our like future like analysis, we wanted to use the longitudinal properties of the MGI of the lab test result as like the longitudinal in the format to validate this kind of causal relationship. Causal relationship beyond like the propensity score or other causal methods. Yes.
Great. Thank you very much. And thank you to all of our speakers, and we'll turn it over to the next session.